Hello, today we are talking about developmental networks of genes. Here is a hypothetical network regarding thorny dragons. The letters represent genes in their DNA, and the protein produced from them reacts with the other genes, either blocking their transcription or stimulating it. The easiest way to understand it is by looking at the arrows as an open door that allows you to easily go through or be transcribed. The T-bars are like locked doors. They prevent you from engaging with what's on the other side. So if a gene is blocked, it won't be transcribed. So if we look back at the developmental network, we have open doors stimulation between genes S and P, K and E produces spikes. However, between P and I and K, we have locked doors, blockage. Let's take a look at some different cells and see whether or not they can produce spikes. This is a wild type cell, so all genes have the possibility of being produced. We have S, which stimulates P. I will be blocked by P, which will prevent K from being blocked. K stimulates E, and produces spikes. Our first mutant cell is unable to produce P. Anything before that, in this case, just gene S, won't matter. We don't have P, so I cannot be blocked. This blocks K. Without K, we can't produce E, and so we cannot produce spikes. This mutant cell doesn't produce I. Again, anything before that won't matter. So we don't have I, so we can't block K. This stimulates E, and consequently, spikes are produced. This is a double mutant cell that doesn't produce I or E. Since E shows up later in the network, anything before it won't matter. We don't have E, so we can't have spikes. Another double mutant now for S and K. K shows up later in the network, so we start analyzing from there. If we don't have K, we can't stimulate E and can't produce spikes. You might be asking yourself, how can different cells express different genes if they're all present in all cells? One hypothesis is RNA splicing. Let's assume all those genes are on the same chromosome. After transcription, the mRNA must be spliced and prepared to leave the nucleus and produce proteins. This process alone can create different mature mRNAs in different cells, and that leads to different proteins being produced. That is why not all skin cells produce spikes.